Hi, my name is Brian Silver, and I'm a fourth-year postdoctoral researcher at the NIEHS, co-mentored by Dr. Tokar of DTT and Dr. Garish of DIR. We use organoids to study biomarkers of disease and toxicity. I hope you enjoy this presentation on what are brain organoids.
Growing miniature brain-like structures in the lab sounds like science fiction, but brain organoids are a real technology being used in research right now. What exactly is a brain organoid? And what's the purpose of trying to grow things resembling features of organs in the lab? Diseases like brain cancer, Alzheimer's, dementia, and other neurological disorders are essentially caused when something goes wrong with the cells in your brain. A big area of health-related research is studying how cells grow and develop, and what types of things can happen to mess up how cells are growing. One method of studying cells in the lab is to examine cells growing in a petri dish in culture. For example, we can expose the cells to drugs or chemicals and see how they respond, or study how specific gene mutations impact cellular growth and behavior. Although this is a valuable way of learning more about cells in the lab, this method has some drawbacks. Your brain, for example, is a complex three-dimensional organ with layers of cells stacked and arranged around each other in complex shapes. Cells in a petri dish are usually grown in a single layer. Also, cells don't just respond to changes in chemicals like exposure to a toxin, for example. Cells are also deeply influenced by their physical surroundings. To understand how physical surroundings affect cells, imagine how your movement and behavior might change if you were squeezed into a tiny crowded room versus being all alone in a large area, for example. In addition to this, organs like your brain aren't made up of just one type of cells. Your brain consists of multiple different cell types like neurons, astrocytes, and glia, to name a few. Organoids are a tool to help us more closely mimic the complex environments and structures of organs in the lab. Organoids are three-dimensional groups of thousands of cells organized into more complex patterns containing several different cell types. We can think of a brain organoid as a miniature, very simplistic model of some of the structures and cell types you'd find in the actual human brain, but it's important to remember that brain organoids don't have anywhere near the complexity found in your brain. Still, brain organoids are more complex and more closely mimic the conditions found in the human brain than a simple single layer of cells. And organoids allow us to study cells that are genetically human, which is an advantage over using research animals like mice. So how are brain organoids made in the lab? The human body is made up of lots of different types of cells that are organized into many different types of organs, like your kidneys, lungs, heart, and brain. But if we think back to the earliest stages of development, way back when you first started out as an embryo, you didn't have all these organs and cell types yet. You were sort of a ball made up of very similar cells. These cells, though, had the potential to turn into different types of cells under the right conditions, a process called differentiation. These special cells that can turn into virtually any cell type are called pluripotent. In the lab, we can take pluripotent stem cells and tweak their growth conditions to trigger them to start forming more brain-like structures. In the case of brain organoids, this is a long procedure, lasting over a month. During this time, human pluripotent stem cells are clustered into groups of cells and then sequentially treated with different combinations of chemicals. After about a week, the now bigger clusters of cells are embedded in a jelly-like substance that changes the physical environment of the cells, guiding their differentiation closer to cells found in the brain. A few weeks later, the organoids grow up to 1 millimeter in diameter and contain several different cell types found in the brain, including neuronal cells and astrocytes. Brain organoids also mimic some key brain-related features, including electrical activity and neural networks. Of course, these activities are very simplified compared to an actual human brain. For example, lab-grown organoids aren't capable of higher thought processes or consciousness. But brain organoids are useful tools for studying brain-related diseases, including neurodegenerative diseases, brain cancers, viral infections, toxicity, and developmental disorders like Down syndrome. Although there are still challenges to overcome like time, cost, and reproducibility, continued research in the field of brain organoids could help us decode the workings of the human brain and improve treatments for brain-related disease.